Hi everyone, welcome back. So this week we are going to get into chapter 5, which is basically we're going to start focusing on the function of some of these organelles we learned about last week. So we're going to start with looking at the plasma membrane and understand how important it really is. So that phospholipid bilayer is what we call a semi-permeable membrane. So only certain things can enter the cell and leave the cell. So this chapter is going to talk about how things can enter and leave while others cannot. Okay, so we're going to get into that this week. So let's get into, so basically this chapter is divided into 5 segments. So we're going to talk about the structure of the cell membrane, and what are the components of it and everything. And then we're going to get into passive transport and active transport, which is looking at the concentration gradients, and we'll get on to all that. And then we'll see things like how big items enter the cell and leave the cell through bulk transport, and then we'll talk about how cells can communicate with each other. So let's start out with a video. So the Amoeba Sisters video will give you a good introduction into what we're going to be studying here.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means 2 layers, so you have these 2 layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass, Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or penocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit, they sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. All cells have membranes, but not all cells have a wall, but plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so I like that video to give us a good introduction into some of the terms that we're going to be using in this chapter, like simple diffusion versus facilitated diffusion versus active transport and those concentration gradients and all that fun stuff. So let's get right into it. So basically, again, we're going to be focusing on that plasma membrane. Now remember that plasma membrane is found in all cells. Okay, it doesn't matter if we're talking about the prokaryotic cells or the eukaryotic cells, all cells have that. And basically its job is to separate the internal components of the cell from the external environment, okay, so from its surroundings. And because it's this barrier, the cell still needs to have certain things come in and certain things to come out, but we only want specific things coming in. We don't want everything flooding in there at the same time. We don't want to open the gates and allow everybody in. So it exhibits what we call selective permeability. So that means that some substances can cross more easily than others. Thank you, Neville. So let's start out by looking at that cell membrane itself. Now, we already talked about 2 weeks ago about the phospholipid, what a phospholipid is. So it's classified as a lipid, and it's one of the most abundant lipids around because it makes up the cell membranes of every single cell. And phospholipids, they're amphithetic, which basically means that they have the head region is hydrophilic, because that's the phosphate end, and then the tail region, the fatty acid area, is going to be hydrophobic. So because of this structure, okay, we have a hydrophilic head and a hydrophobic tail. So that means that we have this interesting bilayer form where we have 2 layers of phospholipids are arranged so that the hydrophobic ends are facing inward and the hydrophilic heads are facing outward. So you can see how we have, here's one bilayer. Let me get my pen out. Okay, so here's one of the phospholipid bilayers, and then here's the other one. So you can see how the tails are facing inward to form this hydrophobic region of the cell membrane, and then the heads are facing outward, which are the hydrophilic regions of the membrane. Okay. So as far as the cell membrane goes. So cellular membranes are fluid mosaics of the lipids and the proteins. So we call that phospholipid bilayer a fluid mosaic model. And the reason being is that it's not just the phospholipid bilayer that makes up the cell membrane. There's all sorts of other stuff. You'll see those proteins, like in the video, the proteins embedded within the plasma membrane, and then even on the surface of the plasma membrane we might have like glycoproteins, or we might have glycolipids and different components at the surface of the plasma membrane. So that's why this plasma membrane is considered a fluid mosaic model, because it's mosaic of different components. It's not just made of the phospholipids. So we have proteins and other things in there and protruding from it. And then when we look at it, we'll see how those proteins and lipids, certain lipids, they form these little patches, which is why it's that mosaic pattern. Okay, so let's take a closer look at our plasma membrane so we know what we're talking about. Okay, so here is a schematic of a plasma membrane. So you can see, first of all, we have that phospholipid bilayer. So you can see how that's just the 2 layers. But then within the phospholipid bilayer, we have these little units. Okay, that is cholesterol. And cholesterol is there to help stabilize the plasma membrane, giving it a little bit more stability and structure to it because it's a little bit more solid. So we have that, and then you can see within the plasma membrane we have embedded different proteins. Okay, so these are different proteins embedded there. And then on the surface we might have other components, like you see here, says glycolipids sticking out. We have these little carbohydrates and these glycoproteins sticking out. Okay, so, and even on the inside we have peripheral proteins and things like that as well. So this is our fluid mosaic model. We have the phospholipid bilayer, but we have all sorts of patterns and mosaics of other things there. Okay, so let's talk about that, the fluidity of the membrane. So we know that the phospholipid bilayer is going to be, somewhat fluid in nature. So most of the lipids and some of the proteins in the membrane do have that ability to kind of shift sideways. This movement of phospholipids tends to be rapid, but the proteins tend to be more slow. And this comes into play when we're talking about things that enter and leave the cell through the plasma membrane. So we'll see how in something like in the video, it talked about endocytosis and exocytosis. Okay, so endocytosis, remember we have some big particle entering the cell, and you can notice that it surrounds itself with the plasma membrane. So that is causing, so the plasma membrane is shifting quite quickly, whereas the proteins themselves are not. They tend to be more anchored in place. They might drift slowly. Okay. So here you can see kind of an example of that. So if we have, if we're trying to hybridize a mouse cell and a human cell, and there's all these member proteins. Once we hybridize, eventually the proteins will start to scatter, okay, but they're a little bit slower. And then as far as some of just the general nature of the membrane itself, remember that we have, as temperature cools, molecules tend to slow down. And so same thing with the membrane. So the membrane can switch from kind of a fluid state to a solid state if the temperature drops too much, which can, contribute to things like, frostbite in your fingertips and so forth. So it's just because things are solidifying, and once it's solid, it's pretty damaging to the tissue. The temperature at which a membrane solidifies depends on the type of the lipids it contains. Membrane remains fluid to a lower temperature if it's rich in phospholipids with unsaturated hydrocarbon tails. So this is important when we're talking about some certain types of cells that can resist those cold temperatures. Okay, so they tend to have these, more phospholipids instead, and less of the proteins and things like that. Okay. So that could be useful in really extreme environments that, some fish live in very cold temperatures and things like that, and they can survive just fine. All right, and then as far as the cholesterol, we talked about how there's cholesterol kind of embedded in the cell membrane, so that it has it's more of a solid form in a warmer temperature, so it tends to keep the cell membrane more stable. So instead of just being this gelatinous mush during the warmer temperatures, so it's going to keep the membrane stable even though the temperature is pretty warm. And then the cooler temperatures, it helps prevent everything in the membrane from tightly packing together. So we want the membrane to be fluid, semi-fluid, right? So we want it to usually be as fluid as olive oil, so a little bit fluid, but not, completely like water. So this is just showing what we want it to be. So we don't want the plasma membrane to be so fluid that, the phospholipids are so kind of far apart, and then we have these little kinks forming. So we want it to be more viscous, where we have these saturated hydrocarbon tails that are pressed together. And then here we see that cholesterol embedded inside that kind of spreads some of it apart, so it's still a semi-fluid, but not completely out of control. Okay. All right. So that's the phospholipid part. Let's talk about those other components in our plasma membrane. So we have the proteins that are embedded in the plasma membrane itself. And a lot of times these proteins are involved with a number of different functions, specific functions. So we might have, let me just go back a couple slides. So here we have we're talking about these big proteins, right? Some proteins we call the integral proteins. So you can see how the integral proteins pretty much go through the plasma membrane. And then we have what are these called the peripheral proteins that are kind of on the ends of the well, in this case, it's on the end of an integral protein, but it's just outside the plasma membrane, so it's on the surface. Okay, so peripheral, think surface. Integral, think in between. Okay, so that's just so that we can talk about it here. So integral proteins, the in-between, so they penetrate the hydrophobic interior, those tails of the lipid bilayer. So they're going to be involved with that transmembrane process where they're going to allow certain things to enter the cell. So they can add what can be considered like an open channel, so they can allow certain things to enter the cell. The peripheral proteins, they are loosely bounded on the surface. So their job is more of like a signaling protein. So here's just an example of an integral protein. So you can see, remember the back in Unit 2, we talked about the structure of a protein. So we have that primary structure, the secondary structure, the tertiary structure, and then perhaps the quaternary. But here you can really see, look what's going on with that secondary structure. We have that alpha helix, and then you can see how it's folded. You don't need to memorize any of the N-terminus versus C-terminus sides, but you can see the complexity of these integral proteins. Okay. So let's talk about some of the functions of those proteins. So for the integral proteins, again, it can be involved with transport. So think of in that video, we had that facilitated diffusion where molecules that cannot just cross through the plasma membrane on their own can cross through with these channels that these integral proteins form. So they can enter and leave the cell through what we call passive transport, through facilitated diffusion, or active transport, which requires energy. And we'll talk about that later. Enzymatic activity, so remember proteins enzymes are mostly made of protein. Enzymes are those types of proteins that help speed up or catalyze reactions. So they can be in the surface of, in the plasma membrane to help facilitate certain reactions. So you can see here we have an enzyme integral protein that is creating these little docking stations, those active sites, so that it'll speed up the reaction. Signal transduction, so here we have little receptor sites, which the protein is basically containing, and that's going to allow for some reaction to take place within the cell or transport a signal. The cell-to-cell recognition, so that we generally have these receptor sites on the proteins, and that's going to kind of serve as a signal for nearby cells. It could also be helping to form those the anchoring junctions. Okay, so we talked about that as part of the cytoskeleton, but it can also be helped with these gap junctions and tight junctions. And then attachment of the cytoskeleton. So that's the extracellular matrix on the outside of the cell membrane. So they can form as attachment sites for that. All right, so just some information about how we bring in carbohydrates, because we've been talking about the phospholipid bilayer and the proteins, but we can also have carbohydrates involved with that fluid mosaic model. So sometimes those carbohydrates, they're bound to the surface of the plasma membrane, and they're going to be serving as receptor sites, okay, for cells to recognize and bind to each other. They can also be combined with other things, like, for example, a glycolipid. So glyco, think carbohydrate, or sugar, lipid, of course, fatty, something that contains lipids. So it's a carb plus lipid component. And then the glycoproteins, carb plus protein component. And so these molecules, they can also be found at the surface of the plasma membrane. Let's see, carbohydrates on the external side vary among species, individuals, and even cell types of the individuals. So they are very species specific or even cell specific, which makes sense because they can serve as receptor sites. So we got to be very specific when we're talking about certain things attaching to the surface of a cell. All right, and then finally, how do we get more plasma membrane? So we talked about the endomembrane system in last week. So remember how the endomembrane system works, where we have basically the components of the rough ER and the smooth ER are going to be transported from the endoplasmic reticulum to the Golgi apparatus. And how are they transported? Well, that endoplasmic reticulum, the membrane of it, is just going to pinch around it, right? So as it's pinching around it, now we have a transport vesicle that contains those components. And then that transport vesicle is going to fuse with the Golgi apparatus, where the components are then modified and packaged up. And then finally, those components being modified and packaged up will leave the Golgi apparatus by the Golgi apparatus simply pinching off its membrane surrounding the vesicle, or surrounding the components to form a vesicle. And then that vesicle contains basically a phospholipid bilayer, right? So as it's moving towards the plasma membrane, it fuses with that plasma membrane. So as this vesicle is exporting things out of the cell, the plasma membrane is going to be growing a little bit, right? So it's going to, because it's adding in that phospholipid bilayer from the vesicle, okay? And then every time something comes into the cell through endocytosis, the opposite occurs, because the plasma membrane will pinch off around that product to form a transport vesicle. So a little bit piece of that plasma membrane becomes that transport vesicle. So we can see basically how we get more plasma membrane with the secretion of things out of the cell. Okay. All right, so we talked about the cell membrane. Let's talk about its selectively permeable nature to it. So we already talked about how the plasma membrane is mainly that lipid bilayer. So we have the hydrophilic heads and the hydrophobic tails. So the hydrophobic tails prevent certain things from passing through. So we tend to have polar molecules. It's harder for them to get through because of that non-polar region of the hydrophobic interior of that plasma membrane. But if we have a non-polar molecule, it typically can squeeze its way through and cross easily. So think of small compounds like oxygen molecules, so O2, carbon dioxide can pass through easily, and so forth. In addition to that the nonpolar molecules like gases, certain gases being able to cross through, and certain ions and things like that, we have those integral proteins, transport proteins, that allow certain things to pass through the channel. So that's going to allow hydrophilic or polar substances to cross across the membrane, or larger substances. Aquaporins are specialized integral transport proteins that allow water through. And then carrier proteins, they can bind to certain molecules, and they can change the shape of the molecule in a certain way that it can shuttle across. Okay, so let's watch a video kind of showing all of this. Oh, I gotta put my pen away.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
All right. So again, that was kind of, you saw how the plasma membrane has that fluidity to it. So certain things can kind of squeeze through, like occasionally water can get through because of that fluidity. And then you see how the nonpolar molecules like lipids can easily pass through, and gases, whereas the rest of them typically need to go through a some sort of transport protein. Okay, so let's get into what both of the Amoeba Sisters video and this last video kind of talked about, which was diffusion. So what is diffusion? So diffusion is basically the tendency of molecules to spread out evenly into the available space. So typically it's just kind of a random motion where molecules want to spread out and stay kind of spread out. So it can, even though it's kind of random, it tends to go in one direction from high concentration to low concentration. So if we have a lot on one side, eventually, even though they're moving in random directions, they're going to spread out towards the low concentration side until the whole thing is more equalized. Okay, so you can see here's an example of just taking some water and adding some dye in it, just little molecules of some sort of color, a yellow color. And then you can see how it's pretty concentrated in one area, and then there's this semi-permeable membrane, so there's little barriers, but then there's some openings the dye can get through. And so the dye is going to want to go from that high concentration to the low concentration. So it's kind of going in a random movement, but eventually some of them are going to make it through, and then it's going to keep going until it's equalized. And even when it reaches this equilibrium, molecules don't suddenly just stop moving, right? They're continuing to move, but it tends to be a net of equal movement on either side. So we might have this same number on either side. They're just equally going back and forth. So let's watch a video showing that.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Okay. So we can see how, in this case, these little green, whatever they are, they tend to be equal on both inside and outside the cell. So they've, they're continuing to cross back and forth at an equal rate. And then you saw how the carbon dioxide was kind of leaving the cell from more concentrated to less concentrated, and the oxygen was entering the cell, again, to reach that equilibrium. So that means that substances, they tend to diffuse down their concentration gradient. So that means that they're going from high concentration to low concentration. And this, across a membrane, this would be a type of passive transport since no energy is involved. Okay, molecules just have that tendency to go from high concentration to low concentration. Okay, so passive transport, think no energy involved. Active transport, we do need that cellular energy, ATP. Okay, because you'll find out we're going against the concentration gradient. So diffusion is going with the concentration gradient from more concentrated to less concentrated. Okay, so you can see we're going from more concentrated to less concentrated until we reach that equilibrium. We can do this on both sides. We can have some yellow dye on one side of the selectively permeable membrane, and then we can have this purple dye on the other side. And same thing, okay, the yellow dye is going to move from high concentration to low, so you can see how it's moving towards the right. And then the purple dye is going from high concentration to low, so it's going from the left to the right until it's evenly spaced. So let's watch an Aviva Sisters video to kind of talk more about it.
Have you ever heard of the ick? The disease, that is, that can afflict aquarium fish? I will never forget the first time my fish caught ick. Well, it's called ick. It stands for this. So, ick it is. It's actually caused by a protist, a parasitic one. In our ecology video, we talk about parasites and how parasites benefit while causing harm to their host. And in my case, it was all of my beloved fish. Well, okay, technically it affected Gertrude first, but then it spread because it is a contagious disease in fish. It typically causes these white dots. It gets on their fins, on their bodies, on their gills, all over. It can even be lethal if not treated. So it's bad news, and being a guppy enthusiast, I was really worried about all of my guppies. This is not a video about parasites. This is a video about diffusion. So you're probably wondering, how is she going to connect diffusion to this parasitic protist disease? Well, it's because of the treatment. You see, there is a commonly purchased treatment for the disease of ick. One common treatment option contains an anti-parasitic called methylene blue. Its name is not misleading. It is quite blue. The directions on the back of the medication outlined how many drops to add per gallons of water. And I watched and tried to encourage my fish to swim towards the medicine. But they were panicked, maybe because a portion of their water was changing colors, but also probably because of the kid trying to get their attention to swim towards the medicine. But you know what I didn't know then? I didn't need them to swim over there. The blue drops would spread on their own in a process known as diffusion. Diffusion is when the net movement of a substance travels down its concentration gradient. That means it moves from a high concentration to a low concentration. When the methylene blue drops were added into the water, they didn't just sit there. The molecules traveled in the water through diffusion. The high concentration of molecules, which is where they were dropped, spread to areas of low concentration in the water. Pretty soon, the water had a uniformly blue appearance as the molecules eventually are evenly dispersed. This was very important for treating my fish because the treatment reached all of them and they soon got better. Diffusion doesn't just happen in water. For example, it occurs if you spray an air freshener in the air. The net movement of molecules travels from an area of high concentration of the air freshener to a low concentration of the air freshener. Pretty soon, it's likely others would smell the air freshener, even if they were quite a distance away. Now, I want to mention two important points. First, we say net movement because that's the overall movement. But it doesn't mean that the molecules can't move around the other direction, and it doesn't mean that the molecules eventually stop moving either. The molecules are continuously moving, even when equilibrium is reached. It's just the overall movement, the net movement, is from a high concentration to a low concentration until equilibrium is reached. Second, diffusion is a passive transport. That means an input of energy is not needed. We say that passive transport does not require added energy. When we talk about diffusion of molecules in cells, for example, the diffusion of oxygen molecules into a cell, it is classified as passive transport. A concentration gradient is itself a form of potential energy. Passive transport is different from other processes that we detail in our cell transport video, like active transport, where an input of energy is required. And speaking of our cell transport video, we outlined that there is also a type of diffusion called facilitated diffusion. Facilitated diffusion is when molecules still have a net movement of a high concentration to a low concentration, but they may be too large or have other characteristics that prevent them from directly traveling across the selective cell membrane. Therefore, they have to go through a protein channel. It's still diffusion because it's still passive transport and it's still traveling down its concentration gradient. It's just that, in that case, it generally has to go through a protein to get inside. Now, there are factors that can affect the rate of diffusion. We'll mention just a few of them when considering these small molecules here, diffusing in simple diffusion. Distance. The greater the distance that needs to be traveled, the slower the diffusion rate. For example, you could observe a difference in a 5-gallon tank versus a 55-gallon one. Temperature. Okay, really quick, would you think a higher temperature or a lower temperature would increase the diffusion rate, assuming everything else is constant for the point of the comparison? Generally, a higher temperature. A higher temperature generally would mean there is more movement of the molecules, so the diffusion rate is increased. This has to do with energy, so check out the link in the description for more about that. Characteristics of the solvent. For example, is the solvent very dense? That could slow the molecules down and decrease the diffusion rate. Characteristics of the molecules, or whatever substance is traveling through diffusion, since technically not everything that travels in diffusion is defined as a molecule. What is the mass of the substance traveling? Generally, a substance with greater mass will have a lower diffusion rate when compared to a substance with less mass. Characteristics of the barrier, if diffusion is crossing a type of barrier. To explain that, let's consider diffusion across a cell, where the barrier would be the cell membrane. Small, nonpolar substances pass through a cell membrane easier than something large or polar. That will affect the rate of diffusion. The surface area and thickness of the cell membrane also will alter the diffusion rate. For example, a large surface area and thin membrane would generally allow a faster diffusion rate than a smaller surface area and thick membrane. By the way, there are more factors that can affect the diffusion rate than just those 5 we listed. In fact, you may have already thought of another factor when seeing our simple diffusion image. Generally, if I were to increase the concentration of the substance here, that would cause a larger difference between these concentrations, and that would also increase the rate of diffusion. Before we end, you might wonder, why should I care about this diffusion thing? Okay, other than the aquarium fish that I love, realize that diffusion is critical for all of life. Cells are the living unit that builds up all life, and materials that cells need to survive have to get in, and waste molecules of the cell have to get out. Now, while diffusion is certainly not the only kind of cell transport, Diffusion is responsible for many critical items. One example, as you sit here right now, diffusion is responsible for oxygen leaving the alveoli of your lungs to your blood. Likewise, the waste gas carbon dioxide is able to exit your blood to the alveoli, a critical process made possible by diffusion. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. So I liked that video's, well, not only it gave us a good overview of diffusion, but it talked about different ways that diffusion rates can change due to things like the molecular size and the distance and the temperature and so forth. So based on all of that information, why is diffusion across a membrane called passive transport? So why do we call it passive? Well, remember, it's going from high concentration to low concentration. It's just the natural law of things, and so it requires no energy. Okay, so it's passive in the sense that you're not exerting any energy for the diffusion of these molecules. So we've talked about passive transport in that video, and it talked about how there was just simple diffusion where nonpolar molecules can simply just pass through the phospholipid membrane. But we can also have the diffusion of water. So the diffusion of water has a special name to it. It's called osmosis. Okay, so in osmosis, we're still having water going from high concentration to low concentration. But this is usually in response to a certain solute that is suspended in that water. Okay, so let me show you what I mean. If you take a look here, look at this U-shaped tube here. Now at the base of this U-shaped tube, there's a selectively permeable membrane. Now this selectively permeable membrane allows water to pass through, but not these green molecules. Okay, these green molecules are just too big to fit through. So on one side of this tube, we have water as well as the solute suspended within it. On the other side of the membrane, we have the same thing, water, but we have a higher concentration of solute. So because there's more solute in this amount of water, okay, that means that technically there's less water, because the ratio is more solute to water. Okay, so we have high solute, low water. Okay, and then on the other side we have less solute. Comparably, there's more on this side, less on that side, so less solute. Which comparatively there would be more water molecules then. Now the solute can't move, right? We cannot suddenly have these green things, make their way through the semipermeable membrane. They're too fat, they're too big to get through. So the only thing that can pass through is water. So osmosis, we're still going from high concentration to low concentration, but we have to pay close attention to what's moving. It's not the solute, it's the water. So water is going from high concentration to low concentration. So we have more water on this side, okay, less solute concentration. So water is going to move from this side of the tube into that side. So that's why you can see the level changing. Okay, so now we have about equal distribution of water to solute on both sides. Okay, so let's watch a video explaining that.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
All right. Okay, so again, that's just showing that, remember, we're always talking about in osmosis, water concentrations going from high to low, okay? So sometimes students get confused because they see a high amount of solute, okay? But we're focusing on the water, which direction the water is moving, because usually the solute can't move.
The plasma membrane.
All right. So again, here's basically showing what is happening exactly at that semi-permeable membrane. So the water molecules can easily fit through. So they're moving from high concentration, you see on this side, to low concentration on the other side. So that's why the water is moving from left to right. Okay, the solutes are too big to move through. So the water has to kind of diffuse through to even the concentrations out. Okay. So here's a checkpoint question for you. Predict the net water movement between two solutions, a 0.5% sucrose solution and a 2% sucrose solution, so separated by a membrane not permeable to sucrose. Okay, so I like to draw a picture. Okay, so here's my semi-permeable membrane. Okay. And then on this side, I have 0.5, sorry, the pen, 0.5% sucrose. I'm just going to do an S for sucrose. And then on this side, I have 2%. Oh, gosh, this pen. It's the mouse. I'm sorry. 2% sucrose. So based on this information, we're not looking at sucrose because sucrose can't move. It's stuck between the, I can't get through. So water is going to be moving. So if I have 0.5% of sucrose on this side, what's the rest of it then? It's got to be water. So what percentage is water here? Of this solution, what percent would be water? So it would be 99.5%, right? Is the water. On this side, we have 2% sucrose. So that means that the other 98% of that solution must be water. Okay, there's nothing else in there. So now we can really figure out the net movement of water, which direction it's going to go, because it always goes from high concentration to low. So we have higher concentration, 99.5% on this side versus lower concentration, 98% on that side. So I know that the sucrose, it's not moving, but the water, where there's, the 0.5% sucrose solution will move in the direction of the 2% sucrose solution. Okay, so it's going to move from this side to that side. All right, so we talked about osmosis. Let's bring it to a little bit more applicable to our human cells or animal cells in general. So animal cells, we don't have a cell wall, right? So we just have our plasma membrane. And water can move from high concentration to low concentration in and out of our plasma membrane. So when we talk about the ability of a cell to either gain water or lose water based on its surrounding environment, we call that tonicity. Okay, so tonicity is basically comparing the concentration of the solute to water in a solution. So if the solution that cell is in is hypertonic, that means that there's high amount of solute, low amount of water that cell is placed inside. So here is an example of a hypertonic solution. So this cell is placed in this solution that has high amount of, I don't know why it's not letting me do anything now. Oh, great. Sorry about that, the program just crashed. Okay, so I've got it back up running. Okay, so let's see if my pen works now. So we're talking about hypertonic solutions. So think high amount of solute, not water, solute. So we have high solute, low water outside of the cell. So since we have high amount of solute and low amount of water outside the cell, the water inside the cell is at a higher concentration. So what's going to happen to that water inside the cell? It's going to go from high concentration to low concentration. So it's going to leave the cell, causing the cell to shrink. This is also known as crenate. Crenate. Or shriveled. Okay, but crenation is the technical term. Which, hey, wait, no, you haven't seen that in lab yet. And then hypotonic, think low solute. Okay, so in a hypo, we have low solute, so that means we have high amount of water. So if the cell is placed in an environment where there's more water on the outside of the cell than inside, then the water is going to want to move from outside of the cell into the cell, from higher concentration to lower. So what ends up happening is so much water enters the cell, and the cell has no cell wall to protect it from absorbing too much, so the cell can burst. And when it bursts, it's called lysed. Kind of like, hey, wait, remember when we talked about hydrolysis? So hydrolysis, hydrolysis, water breaking. So think lyse as, lysis, breaking. So the water is entering the cell and it's broken open the cell because there's too much water inside. Okay. And then finally, the last type of solution the cells can be placed in is called isotonic solution. So think iso, equal. So equal means that there's equal concentrations inside the cell as outside. So here's an example of a normal red blood cell in an isotonic solution. So there's equal amount of water inside the cell as outside. So the water will move into and out of the cell at an equal rate. Okay. So when you're in the hospital receiving, fluids, intravenous fluids, the fluid solution should be an isotonic solution. Okay, so it should have some solutes in there because your blood has solutes in there. So as it's injecting that fluid into your body, we don't want a hypotonic solution because then it's just going to cause all your cells to lose water. Basically burst. If we add a hypertonic solution to your bloodstream, it's going to cause those red blood cells to shrivel and crenate. Okay. All right. With regards to plants are a little different because of they have a cell wall. So because they have a cell wall, it does prevent them from absorbing too much water, okay, or losing too much water. But they can still be placed in a hypertonic solution and a hypotonic solution and see some changes take place. So let's take a look at these little videos. When I press play, you're going to see something happening inside the cell. So here we have a plant cell. And in this plant cell, what I want you to notice is, remember, right up on the inside of the cell wall will be the cell membrane. And these little green things are the chloroplasts. So see what happens, pay close attention to the cell membrane when I press play. Okay, so here's the cell wall, just on the inside of the cell wall is the cell membrane. Okay. So look at what's happening. Look at how the cell membrane is pulling away from the cell wall. And basically you're seeing like all the green chloroplasts kind of concentrating. So in this case, the cell membrane is being pulled away. So that means that water is leaving the cell. So was this plant placed in a hypertonic or a hypotonic solution to cause the water to leave the cell? So that would be an example of hypertonic. So one easy way to remember that is just remember that when you can see the cell membrane really well, it's in a hypertonic solution. Otherwise, it's usually just right up against the cell wall and you can't distinguish the two. Okay, so if you can see a cell membrane, it's in a hypertonic solution. So that must mean over here we have a hypotonic solution, which basically you cannot see any cell membrane. It is pushed right up against the outside of the cell or the cell wall. And you can see even, look at where the chloroplasts are. They're not all in the middle. They're kind of all over the side. Okay, so that cell membrane is pushed right up against that cell wall. So that's a hypotonic. All right. So hypotonic is actually kind of the condition plants want to be, because they can't absorb so much water that they'll burst like an animal cell because of the cell wall. But you know, they have lots of water, so they can survive a pretty long time now if there's no water available. All right. So here's just some more information about how the tonicity is a little bit different reaction in plant cells versus animal cells. So cell walls for the plant cell have that ability to help maintain the water balance. If a plant is placed in that hypotonic solution, the water will enter the cell and cause the cell membrane or the cell to swell until it reaches the cell wall. Okay, so we call that a turgid state, or a very firm state, because it's right up against the cell wall. If it's in an isotonic state, that means there's kind of equal amounts. The cell membrane can kind of pull away a little bit from, it's, you know, not firmly up against the cell wall, so it's considered flaccid. Okay, and that, you know, we might see that the plant, because it's kind of limp, the cells are a little limper, the plant might be a little limper, it might wilt a little. And then hypertonic is when we end up with the cell wall is completely pulled away from, or excuse me, the cell membrane is completely pulled away from the cell wall. The water is just, you know, leaving the cell at such a rate that there's not enough water left to maintain the cell. So this could lead to something called plasmolysis, which basically means that the plant is, the cell is dead. There's not enough water left. Okay, and that's when the plant is just bleh, there's no way it's going to come back, or at least those cells won't. Now we talked about how animal cells, they can crenate or lysis, and then plant cells, they can kind of have that turgid versus the flaccid versus the plasmolysis outcome. In unicellular protists, they actually have what's called a contractile vacuole. And contractile vacuoles, we talked about vacuoles before last week. Remember how I briefly mentioned like the central vacuole in plants that's used for storage. Some unicellular protists, they have these things called contractile vacuoles. And these contractile vacuoles are able to aid in what's called osmoregulation. So trying to control the solute balance and the water balance. So this is necessary if you're living in an aquatic environment. Okay, so paramecium, this is an example of paramecium under the microscope. You can see that contractile vacuole, which it's going to use to pump water out when too much water is coming in. So in this case, you know, where do paramecium live? They live in pond water, which is a freshwater environment. So if you have too much, you know, the water concentration is higher outside of this unicellular creature than inside, then water is going to want to come in, right? So that contractile vacuole is useful to pump that excess water out to help maintain, you know, enough of a balance that it can survive. Okay. All right, and then this is just basically a drawing showing the hypotonic, isotonic, and hypertonic solutions with a plant cell. So remember the hypotonic, that's when it reaches that turgid phase where the cell membrane is pressed right up against the cell wall. And this is its normal phase. This is the phase the cell wants to be in, because it's got a nice full central vacuole. It's got plenty of you know, water to sustain it. Flaccid would mean that there's an isotonic solution, so it's kind of equal. So water is just kind of moving in and out of the cell at an equal rate, which allows you to kind of see that cell membrane now, and that can cause the plant to wilt a little because it's more flaccid. And then here's hypertonic, so you can see what happened here. Well, the cell membrane really pulled away. There's no water left, hardly so much has left the cell that it's plasmalized it. All right, so we talked, that was all simple diffusion and osmosis. So let's get into facilitated diffusion. So facilitated diffusion, remember, usually we have some sort of transport protein that has an open channel that allows solutes to enter from high concentration to low concentration. It doesn't always have to be solutes, however. We could have an aquaporin where we have water just moving through from high to low instead of just slowly making its way through the plasma membrane. Or we can have ions. So an ion, we might have certain ions entering or leaving the cell with its concentration gradient based on some sort of gated channel that will open due to a stimulus. And then the Immunosisters video talked about those carrier proteins where it will change the shape in order for something to pass through. Okay, so let's take a closer look at these carrier proteins. So here is an example of a carrier protein. So in a carrier protein, you have, in this case, some sort of molecule. There's high concentration on the outside. And as it enters, it's going to kind of change the shape of the protein. So you can see how it changes the shape. So now that whatever the solute is passed through. Okay. And again, we're moving from high concentration to low concentration. So we don't have any energy. This is a form of passive transport, specifically facilitated diffusion. So how do transport proteins contribute to the membrane selective permeability? So remember, transport proteins allow basically things that cannot pass through the plasma membrane normally. It can help promote their movement. So we can have the polar molecules, we can have larger molecules, we can have things like that, ions, certain ions pass through. So it's still selectively permeable, only certain things can pass through. All right, so we've talked about passive transport, so in the sense of simple diffusion and facilitated diffusion and osmosis. Now we're going to take a closer look at going against the concentration gradient. So this is where we have something called active transport taking place. So we know that facilitated diffusion, we can have substances go from high concentration to low concentration. But sometimes we need the opposite direction to occur. We need solutes that are maybe inside the cell that we don't want them in there. We want to push them out, even though there's lots of them outside. So we want to move certain things against their concentration gradient. So instead of going from high to low, we want them to go from low to already high. And because of that, we're going to need some energy. Okay, so this is just summarizing passive transport. So we're going from high concentration to low concentration. Active transport, we're going the opposite. So here you can see we have very little of these little circles, okay, and we want them to leave the cell. We have very little of these squares on the outside, but we want them to enter the cell at a higher concentration. Okay. So in order to do that, what do we need? ATP, energy. So active transport, we're going to move the solute against their concentration gradients from low solute concentration to high solute concentration. And in order to do that, we're going to have to require some energy. Okay, so energy is going to be used to go against that concentration gradient. So here's a question for you. Cells actively transport calcium ions out of the cell. Is calcium more concentrated inside or outside of the cell? Okay. So here you have to pay close attention to the words. Cells actively transport calcium out of the cell. So if we're actively transporting, that means that we're going from low concentration to high concentration. So if we're actively transporting calcium out of the cell, is calcium going to be more concentrated in the cell or outside? So it's going to be more concentrated outside the cell, okay, because we're pushing more outside of the cell against the concentration gradient. So there's more already outside than inside. So here's a famous example that always is used when we talk about active transport, and even the Amoeba Sisters video alluded to it, the sodium-potassium pump. So this is something that is required for us to get information from our brain all the way down to our toes through our nervous system, through neurons. And basically, this is an example of active transport with sodium ions going against their concentration gradient, going from high concentration outside the cell to low concentration inside. And then the potassium ions being at high concentration inside, we're pushing them against their concentration gradient to go the opposite. So here we see we have low amount of sodium inside, high amount of potassium inside. So right in this situation, we have low sodium, high sodium, and we're going to be pumping more sodium out of the cell. So you can see how that is occurring with the action of ATP. So now the sodium is released from the cell, where we still have more sodium outside than inside. And the same thing is going to occur with the potassium. We already have a high concentration of potassium inside the cell, but we're going to get even more in. So we're adding even more in through active transport. So this sodium-potassium pump works for two different ions. Both of them are going against their concentration gradients. So we're moving the sodium from low to high, inside the cell to outside. We're moving the potassium, there it is, potassium from low outside the cell to high inside the cell.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here's how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
All right, so again, you can see that the sodium is going from from low concentration to high concentration. So it's inside the cell and it's moving more to even the higher concentration side. Whereas the potassium is going from low concentration to high concentration as well, going from outside the cell into the already higher concentration inside the cell. Okay, so in addition to just the transport of different sodium ions and potassium ions, we can also talk about, instead of just looking at a chemical concentration gradient, we can talk about a membrane potential, which is dealing with the voltage across a membrane. So voltage is sometimes it's necessary to balance the amount of positive and negative ions across a membrane. And so that can also trigger a response in the selective permeability of a membrane. So when they're combined together, when we're combining a chemical force, which is the ion's chemical gradient, with the electrical force, looking at the positive or negative of that ion, we get something called an electrochemical gradient. So that electrochemical gradient can then be used to diffuse ions across a membrane. So this, we're going to kind of look at this in relation to, again, the sodium-potassium pump, in addition to, looking at sodium and potassium. So let me go ahead and show you an example. So, hey, look at this funny looking thing. That's a neuron. So neurons are really, strange looking cells. This is one single cell. The nucleus is in what we call the head of the neuron. And then you can see these are, these little finger-like projections are called dendrites. And then we have the axon, which is like the tail-like structure. But let's take a closer look at that tail-like structure, that axon. So here is what it looks like, very simplified view of the axon. So we have our plasma membrane, okay? And then we have the inside of the cell and the outside of the cell. So when we are going from, let me go ahead and take a closer look. So here's our plasma membrane of one side. We're just looking at, maybe just this area right here. And you can see that we have some regular transport channels open. Okay, so in this regular transport channel, we just have facilitated diffusion taking place. So remember, facilitated diffusion, no energy required. And we have the potassium ions going from high concentration to low concentration. We have the sodium ions going from high concentration to low concentration. Now look at what's happening here. We have one sodium channel to two potassium channels just in this little segment. And when they're open, we're going to end up with more of the potassium passing through at a quicker rate. Okay, so we're going to end up with more positive charges outside of the cell because there's, Na is a ion is a positive charge, and so is sodium. So we're getting lots of sodium, we have already a lots amount of sodium outside. Not as much as it's slowly diffusing across inside the cell, but we have a much faster rate of potassium being added to the outside. So we end up with more positives outside the cell. So that means there's less positives outside the cell. So it's almost like a more negative charge. So it's more negative outside the cell when these channels are open. Okay? Now, having these channels open, you can see we have a positive on the outside and a negative on the inside. We can have some sort of electrical impulse take place where suddenly the gradient shifts. So we start seeing the closure of these channels. So if we have a closure of these channels, and the trigger of this one to take place, now suddenly we start seeing, some sodium going out, but we're going to see lots of potassium going in. In fact, there's, I think there's like 3 potassiums go in for every 2 sodiums that go out. So that's going to change the voltage of the membrane. So we're going to start seeing more negative on the outside, more positive on the inside. As the sodium-potassium pump kind of gets turned on. And so you can see that, we have a combination of chemical gradients, high concentration, low concentration, but we also have electrical gradients taking place here. So I'm not a good drawer. Amoeba Sisters video, it is. Okay.
I've always been into fish. And if you're into fish, you're also going to be into fish tanks because you have to house your fish. 5-gallon, 10-gallon, 20-gallon, 55-gallon. These are all tanks I've owned. And with all these tanks, I've had pumps. Pumps are important because whether they are helping to aerate the water or bring water through a filter, they help your fish live their best fish life. The pumps I've always had plug into an electrical outlet and they work hard drawing air or water through some type of tubing. You have pumps in your cells too. Tiny, tiny little pumps because, well, your cells themselves are microscopic. And while these pumps don't plug into electrical outlets, many types of pumps do require an input of energy to power them. ATP is a great example of an energy currency used to power many of these pumps. And we have a video on that. And in our ATP video, we state one of our favorite pumps, the sodium-potassium pump. We also mentioned in that video that we might need to make a video on that pump. This is that video. Now, the sodium-potassium pump isn't trying to aerate your cells, so while it has the word pump, it really isn't much like a fish tank pump. But it is requiring an input of ATP energy, as we mentioned. And it is doing something very important. One major function is it is helping to maintain a resting membrane potential. We should explain that. This is an animal cell here. Here is the extracellular part. That means outside of the cell. This is the inside of the cell here. We're going to mention two ions specifically, sodium and potassium. These ions have positive charges and, like most ions, they have trouble traveling directly through the cell membrane. Instead, they need a protein to travel through. The protein could be a channel, it could be a pump, just some examples. So back to this resting potential. This involves its electric potential, the difference between the electrical voltage inside and outside of the cell. Cells have their own resting potential depending on the type of cell. At rest, generally, most cells are more negative inside the cell than outside the cell. For many cell types, when this resting potential is changed by some type of stimulus, there is a response. Muscle and neuron cells, for example, are excitable cells, and many of their functions depend on a change to their electric potential. We're not going to go into the change of a cell's electric potential in this video. Explore the action potential if you're curious about that. Instead, we want to talk about how the resting potential is maintained. How is it more negative inside a cell versus the outside of a cell? For a cell at rest, or if it needs to return to its resting potential, the sodium-potassium pump is pretty critical. The sodium-potassium pump is initially open on the intracellular side of the cell. It has binding room for sodium ions, 3 specifically. When these 3 sodium ions bind, the protein is phosphorylated. In our ATP video, we talk about what that means. ATP, a molecule of energy currency, transfers its phosphate to the protein. This actually changes the protein's configuration, the protein's shape. This shape change causes the pump to open to the outside. The sodium ions are pushed out to the outside, the extracellular side, and now, due to this configuration change, it's open to the extracellular side. It has 2 binding sites for potassium ions. 2 potassium ions bind. After the potassium ions bind, the phosphate group is released from the protein. The sodium-potassium pump reverts back to its original position, so it will open once again to the intracellular side. The potassium is dropped off inside the cell, and once again, there's room now for 3 sodium ions. The cycle repeats. We should mention that this pump is moving sodium and potassium against their concentration gradient. What do I mean by that? It keeps putting more sodium outside, where there is already a high concentration of sodium. And it keeps putting more potassium inside, where there is already a high concentration of potassium. So with this active transport, the ions are moving from low concentration to high concentration. A great example of active transport. So what is the result of this pump's activity? Well, you are moving 3 positive ions out for every 2 positive ions in. So while this may contribute to the cell's more negative charge inside, it's really important to understand that this alone is not the big reason you see a voltage difference in the resting potential. The result of the pump's work means that there are specifically more potassium ions inside the cell and more sodium ions outside of the cell. This causes an electrochemical gradient. That means there's not just a difference in charge, but also in the actual ion types. Remember, in our diffusion video, we talk about how substances have a net movement from a high concentration to a low concentration. Well, if going by the chemical gradient here, those potassium ions inside the cell, they'd like to have a net movement out. They generally can't travel through the membrane directly themselves, as we mentioned, but they could if there happened to be a channel. These channels do exist. It is common for many cells to have far more potassium leakage channels than sodium leakage channels. This makes a cell membrane more permeable to potassium at rest. So while sodium would like to diffuse in, there are less leakage channels for it to do so. Positive potassium ions that travel out of the cell contribute to a more negative charge inside the cell. If really exploring resting potential, there are additional factors that contribute to the cell's more negative charge. Keep in mind also, because this video is focused on the sodium-potassium pump, we focused on the ions, sodium and potassium. But there are other ions that can contribute to the resting potential as well. And now this electrochemical gradient is a perfect setup for a whole lot of things. We already mentioned that an action potential, which makes a change to the cell's voltage, could result in a cellular response. We should also mention that there are other proteins that rely on this gradient to move critical molecules. Some proteins that transport in glucose, for example, require that gradient to be established. Overall, the sodium-potassium pump is one pretty remarkable protein pump. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Solutes here. We have active transport occurring with one solute, and because that active transport is opening up this little transport channel, we can also have just a facilitated diffusion of another solute along with it. So in this case, we have sucrose. Sucrose is going from low concentration to high concentration. So this is the active transport part. But because the channel is open, it's inadvertently allowing this diffusion of hydrogen ions. So these hydrogen ions are high concentration to low. So they're being transported along with the sucrose through co-transport. Okay. So let's go ahead and stop right here, and then we'll pick this up in the next lecture.